Hi guys, can you hear me?
Yes. Yes, sir.
Thank you. So what are we? We are working on chapter 4. Chapter 4, Monday, September 21st. And we are 6 days before the test, right? When is the test? 20, no, 30, no, not 6 days, or 9 days. You still have time. So hopefully you have had the chance to look at the practice test. And as I mentioned to you, if you have any questions, just throw them into the discussion section. Are there any other questions regarding the class? Any general questions?
The practice test isn't a grade, right?
The practice test is what?
It's not a grade.
No, you can take it as many times as you want. It's just the example of the questions you will see on the actual test. Yeah. And it's for you kind of to see where you stand. If you can answer them, that's great. Means you're all set for the actual test. So, okay. All right, I'm gonna go and share the screen. Let's see where we are with the sorry, my cat is not happy. Hey, quiet. So we have been discussing collagen fibrils, silk fibrin. Right, so we started talking about globular proteins. Globular proteins. So let's continue our discussion of those. So remember, fibrous and globular are the two types of proteins we are talking about. And so globular is the combination of secondary structures which are put together into a tertiary structure. You can see the whole bunch of alpha helices here, right? So this is the structure of the protein myoglobin. So it's a water-soluble protein. You might imagine that actually in the, it's not shown here, but from the knowledge that you already gained in this class, you should be able to predict that in myoglobin, like for example, on the outer surface of this alpha helix, right, which is exposed to the aqueous environment, you will find a lot of polar residues, serine, threonine, cysteine, lysine, glutamate, glutamine, and so on and so forth, right? On the inner surface, the surface that actually is facing inside the protein, you will find a lot of hydrophobic residues, right? Alanine, phenylalanine, valine, leucine, isoleucine, and so on and so forth, right? So, but just by looking at the structure, at the tertiary structure of a protein, you already know how nature designs things, right? You already know that if this protein is water soluble, you already know how nature will put the amino acids on these alpha helices. And you can see here that by putting these three-dimensional structure to these tertiary structure together, just for example, if this was all in beta conformation, these will be the dimensions of the protein of the myoglobin. If this was all of alpha helix, these will be the dimensions. But nature manages to make it so small that it's so globular, it's almost like spherical. So it's almost like it's 100 by 60 angstroms, right? So it becomes a very small protein, right? Because of the compact nature. All right, so this is something that we need to also remember. So this is its high level of structure. So we know what primary structure, we know secondary structure, we know tertiary structure. Before we move on to quaternary structure, there is also something called motifs or folds, most commonly motifs. motifs, basically, these are structural specific arrangements of secondary structure elements of alpha helix and beta sheets. And so these can be found as recurrent structures in numerous proteins. All right, so for example, so beta alpha beta loop. So now this is a very recurrent, this is a commonly recurring motif in protein structures when you have alpha helix, right? And then you have a parallel beta conformation, right? So very common. Now, if you have just the beta conformation in the protein, right, for a long segment of that, it can form what's known as a beta barrel. Beta barrel. So you can see here, this goes in this direction, this goes in that direction, this direction, this direction. So this is an anti-parallel beta conformation motif, right? So it's capable of generating this beta barrel, highly likely. Now, there's if you have these beta alpha beta loop, right? If you have a whole bunch of them, of these motifs, they can aggregate further to form what's known as alpha beta barrel. Alpha beta barrel, and there's actually very nice, it's a very common among proteins which accept various drugs. Like this area here is very hydrophobic. And so various drugs and small molecules can get inside. All right, and the final structure we're going to briefly mention is that, well, it's not a structure, it's actually intrinsically disordered proteins. So proteins whose amino acid makeup is such that they don't really benefit from any kind of secondary structure. Alpha helix, no. Beta sheet, no. Beta turn, no. And so it turns out that these proteins just like to be disordered. And so they're intrinsically disordered. And they're usually composed of amino acids whose high concentration forces less defined structure. So usually if you will see a lot of lysines, arginines, glutamines, and prolines, right? So prolines, remember, they don't like anything. So if you have lysines, arginines, and glutamates, so these are charged amino acids that will want to be on the outside of the helix, right? Forcing all the hydrophobic stuff inside of the helix. And so that hydrophobic stuff inside of the helix, because there's such tremendous force driving those hydrophobic stuff among the helix, the hydrophobic amino acids don't adopt any particular structure. And so basically with the intrinsically disordered proteins, all you have is just like a some kind of sphere. You have pluses, minuses on the outside, right? And here you will have hydrophobic. Without any kind of definable structure. So these kind of proteins will be intrinsically disordered, right? And so what's the, you might wonder what's the advantage of those? Well, they can conform to many different proteins, facilitating interaction with numerous different protein partners. They are there's a word for it, promiscuous. Promiscuous. Which means they can interact with different protein partners, right? Since they don't have a specific definable structure, they can interact with many proteins and form some kind of bridges, glues, stickers, whatever, can perform different functions between different proteins, right? So which is very important. So intrinsically disordered proteins are a very important type of proteins And there's a lot of study done on those. And it's actually turns out that actually many proteins right now have these kind of regions that are intrinsically disordered. For example, let me actually give you there is a P27. P27. It's a tumor suppressor protein. What's a tumor suppressor protein? Well, I mean, basically, the name really tells you what it does. Tumor suppressor. Basically, what it does, it because of its disordered nature, it wraps around kinases. Remember I told you about kinases and their participation in cell division, right? Phosphorylation kinase cascade. So it wraps around kinases and stop cell division. If they stop cell division, they stop tumor cell proliferation. They stop tumor cell proliferation. They are tumor suppressor. Okay, so P27 is one of them. In fact, many cancer cells try to suppress they try to produce less of this P27 tumor suppressor protein. All right, so because obviously they don't want it. They want to divide, right? They want to divide, and they change their genetic machinery so that the P27 is produced in small amounts, is not expressed as much as in normal cells. And in fact, if you take a biopsy of a tumor from a cancer patient and biopsy of a normal tissue from a non-cancer patient, you will find much more P27 in a normal patient, right? So tumors suppress the production of P27 because they know that P27 will suppress the tumor growth. And in fact, the amount of P27 is actually a can be used as a prognosis, as a what's the word? Survival prognosis, right? So if you find very little P27 in a tumor of a cancer patient, then the survival prognosis is very small, right? So you know that actually that this patient will not live long enough because P27 is not produced in large quantities and tumors will grow very quickly, right? So P27 is a key one. So another one is P57, so P53, and that's the one on the next page. So you can see here, this is the natural predictor of disorder. Okay, so there are computers now. So we have learned some things about intrinsically disordered proteins. And so there are computer programs that you put this primary sequence in, and they can actually, you can see here that actually they can predict which region will be disordered, which one will not be disordered. And so this program at the end spits out at the carboxyl. Now this is the amino residue terminus, right? And this is the carboxyl acid terminus. And it turns out that the P53, so this is the protein, also tumor suppressor. Also tumor suppressor. So it turns out that actually, because of the promiscuity So here in this region, you can adopt different conformations, right? It's a very promiscuous AND of P53. It can actually interact with a bunch of different proteins. Can interact with cyclin A, sirtuin, CBP, bromodomain, and this one. And so because of that, actually it can perform multiple functions. So one advantage of intrinsically disordered proteins is the lack of specificity, right? Lack of specificity is the same as promiscuity, right? So there are plus and minuses. The plus is that obviously it can perform more than one function. The minus is obviously, we always said that proteins, one of the biggest advantages, they're so specific, right? So here we'll lose that specificity, and the P53 can interact with multiple client proteins. Okay, any questions about intrinsically disordered proteins?
So both of the examples we have are tumor suppressors. Is that pretty common, or is that just a coincidence that we covered these two?
I would say it's coincidence. Now there's for the intrinsically disordered proteins, I mean there are many different functions for those. In cancer cells, there's, I would say maybe half coincidence, because cancer cells like change, right? They like to be able to evade the host immune system. They like to be able to evade the chemotherapy. And so they do like to produce intrinsically disordered proteins, because those can change shape and three-dimensional structure and evade various cell defenses against disease, right? So maybe what you're saying is correct. Maybe it's not so much coincidence. It's I don't know. It's just my guys, with science, you can never know everything. And so what this class is meant to do for you is for you to start thinking on your own, right? Just gain enough material, gain enough rules and concepts so that you can start thinking on your own. And if you can come up with the idea why cancer cells overexpress tumor suppressors, great, So you can explain it for yourself. All right. There was something I wanted to write down, but now I can't remember. Something I want to write down. Anything else? All right. And quaternary structure. So let's see how much time we have. 20 minutes. So quaternary structure formed by assembly of individual polypeptides into a larger functional cluster, right? So here we have, so this is the protein known as hemoglobin. Hemoglobin, we'll talk about hemoglobin quite a bit in chapter 5. Oxygen carrier in the cells. Remember, oxygen is not very well water soluble. And so oxygen will bind into these heme prosthetic groups right there. Right there. And hemoglobin will carry them throughout the general circulation into the tissue. And so hemoglobin represents a quaternary structure. And remember, quaternary structure is a structure of a multi-subunit protein. Right, so tertiary structure, multi-subunit protein. So tertiary structure is a combination of secondary structures, right? But the single protein. And now if you have a number of such single proteins combining together, forming a much larger macro scale structure, it will be a quaternary structure, multi-subunit. All right, so we talked about circular dichroism. So I mentioned that to you last time, right? So it's a classical method to deduct the or deduce to deduce the secondary structure, but that's an old one. Now there is a much more advanced method, which is still old, but the advances are in this area have made it very important, is X-ray crystallography, right? So remember, so X-rays, let me find this in the text. So basically these are based by, so electron density, that's what the technique detects. Electron density will diffract X-ray beam. Now, if you look at advantages and disadvantages, can you find on this list the advantage or disadvantage, one of them that will depend on, that will directly depend on this property, that electron density will diffract the X-ray beam. So basically you look for diffraction, right? You look for diffraction, and where the X-ray beam is diffracted, you find an atom. You find an atom. Basically it gives you a picture of atoms. What could be better, right? A photographic picture of all the atoms in your molecule. But it will not give you a photographic picture of one type of atom. Which one is that?
Hydrogen.
So why will it not tell you where hydrogens are?
Because they can't see them.
Why can the X-ray beams not see hydrogens?
Are they too small?
Well, too small, but look, electron density, that's the word, that's the key word. Do hydrogens have any electron density?
Oh, there's no electrons.
There's no electrons, that's right. So most atoms will have S electrons, P electrons, inner shells, outer shells, and so on and so forth. Hydrogens don't, right? So the problem with X-ray is that it will give you the skeleton, the bones, but it will not give you the decoration. It will not give you the hydrogen atoms attached to other atoms, which, and obviously all these programs which will attach them for you, or you will just have to attach the hydrogen atoms manually wherever they're missing. You put them there. So it's not a big deal, but sometimes you would want to see how many hydrogens. So basically it will give you an oxygen, but you have to figure out if there are 2 hydrogens on this oxygen, 1 or 0. But because you know that, you know the valence, right? You know how many valence electrons there are. So you know general chemistry, you know X-ray crystallography. All right, so what do you do? So first you purify the protein. Now you know from OChem 2, right? So you run a reaction. If you need to crystallize, let's say you have a precipitate and you need to crystallize it. If your precipitate is dirty, if it's impure, you will never be able to crystallize it, right? So the impurities will get incorporated into the powder, and you will not be able to crystallize. You need to have pure material. Now, secondly, crystallize the protein. Lots of different techniques. So protein crystallographers, it's a job for life. right? So I've never tried myself, but I know that I will suck at it. You know, it's something that maybe after I've done it for a few months, I can learn a few things here and there. And but in general, to crystallize a protein, it's a skill. It's a skill. So why do you think it's a skill? What's the problem? So you've done crystallization in OChem2, right? You've done crystallizations in OChem2. And so you have a precipitate, right? So you heat the solution, it'll go into the solution, and then you cool it down, it'll crystallize, correct? What's the problem? Where does my logic break?
Can you rephrase the question, please?
Well, rephrase the question. You've recrystallized compounds in OChem2, haven't you?
Mm-hmm.
So how did you do it? So describe me the process. How did you do it? Anybody? Just, you know, it's
Is the issue that you have to heat it up and that could denature the protein?
okay. One very good issue. Very good issue. If you heat it up, it will denature the protein. And we will actually look at the later, hopefully we'll have enough time, at protein denaturation. Yeah, good one.
You have
What else?
to remove the impurity by using a stationary phase and a mobile phase. It's different ways you could go about doing it.
Let's say you can you can heat the protein from 37 degrees Celsius, which is room temperature, to 40. It will go into solution without denaturing. Let's say you manage to do that. But still, what's the problem then?
It still contains the impurity.
It contains the impurity. So remember from organic chemistry 2. So you heated your compound, bring it to boil, right? You bring the solution to boil, and you put it aside for it to crystallize. Did you just put the flask aside, or did you actually do anything to it? What did you do to that flask?
Generally, you put it in an ice bath.
Oh, that would be at the very end, right? But before you, I mean, right after you brought the compound to boil, did you immediately put it in an ice bath? Because see, the problem is if you put it in the ice bath right away, it will precipitate very quickly and form a amorphous powder, not crystals.
You need to let it cool down to room temperature before you put it in the ice bath?
And quickly or slowly?
Slowly.
So why do you need to cool it slowly?
Because the impurities could get trapped
That's
in
right.
the crystallization if you cool it down too fast.
That's right. Remember, crystallization and going back into the solution is a reversible process, right? So when a crystal is growing, when a crystal is growing, you want your molecule to come to the crystal and decide if it wants to be part of this crystal or not. If it comes to the crystal in such a way that it does not fit in there, you don't want it to misfit. You want this molecule to go away and come back in a better orientation. So you want this whole process to be in equilibrium, right? So you want the molecules to equilibrate and find the best place in the crystal. So the crystal, if you remember, the best crystals, the most pure crystals also have the most beautiful shape, right? The purest crystals have the most beautiful shape. And why is that? Is because the compound finds the best position for itself within the crystal. Because the impurities are not intermingled in between the molecules, right? So you want this to equilibrate. So you want to lower the temperature as slowly as possible. So you want the compound to come and decide. You don't want to come. You don't want the compound to come and immediately attach itself to the crystal and form some kind of amorphous, you know, God knows what, precipitate. You want it to be a beautiful crystal that fits, where each molecule fits snugly into its place. Does this make sense? So crystallizing protein is a skill. And so oftentimes what happens, people, instead of temperature, instead of playing with temperature, so as was very nicely mentioned to us, that by playing with temperature, you can denature proteins. Often what people do, they add various alcohols, for example, like methanol. So you have a desiccator, which is a closed container. So you put your protein of interest in the buffer, right? And in one dish, and next to it, you put a dish, let's say with methanol. Put a dish with methanol and put it under vacuum. And methanol molecules, which are much more volatile, will leave the dish, travel into the dish with the protein, dissolve themselves in a dish with the protein in a buffer, lowering the polarity of the buffer, and forcing the protein out of the solution slowly, right? So sometimes crystallization takes days, sometimes takes months. So people who are protein crystallographers, they have lots of patience. And guess what? Difficult for membrane proteins. What is so difficult for membrane proteins? Why is it so difficult to crystallize a membrane protein? What do they have on the outside?
Is it because of the bilayers?
Well, they have hydrophobic residues on the outside, right? Now, in a crystal, remember, to form a nice, strong crystal, you have to have polar residues on the outside, which can interact with each other to form some kind of charged interactions, right? Or dipole interactions, which are stronger than any kind of hydrophobic forces. So a lot of membrane proteins, even though we have known them for dozens of years, they still have not been crystallized. And oftentimes, nowadays, if you read the journals, such as, now, I don't know if any of you read some science journals. You should probably start, right? Many of you are juniors and sophomore, juniors, and seniors, right? So you should start reading science journals. Just maybe like one article per journal or something, just to keep to keep up with what's going on in science. What's the top journal in science? Like which journal? Like if I told you to read one article weekly from a top journals in science field, which journal would you choose? Now, those of you who go to medical school, this is a question that you may be asked very well during your interview. Are you ready for it? Which journal? What's the top journal in science, which describes a lot of breakthroughs in medical field? Which journal would you read? What am I writing? No? What's missing?
Nature.
Nature, good job. How about this one? What am I writing here?
Science.
Science, good job. So Nature is the journal by what's known as the Royal Society of, sorry, take it back. Nature is the British journal. Nature is the British journal, but I mean it's international. Like Science is a journal for American advancement of science. But journals, as you know, their quality is based on what's known as the impact factor. Impact factor is 40 to 50. Do you guys know what impact factor is? So basically, impact factor is the number of times a paper is cited in some other papers. So let's say your journal publishes 10 papers in an issue, right? And so these 10 papers are cited 400 times. Then the impact factor is 40, right? So basically, the number of times papers are cited per each paper in an issue, 40 to 50. So that's extremely high number, 40 to 50. So just to give you an idea, my best paper that I ever published was I've never published in Nature and Science. I'm not a good scientist, I guess, but my best one is in the Journal of American Chemical Society. Its impact factor is 13. And even though I'm very proud of it, very proud. There are more than 50% of all the journals impact factors. More than 50% of all the journals impact factors less than 1. Which means you publish a paper and you have to pray that somebody cites it for you. All right. Anyway, so this is X-ray. This X-ray, we spent quite a bit of time. Let's move on. Let's move on. So yeah, so this is how it's done, even though it's very hard to understand with the naked eye, but X-ray crystallographers, it's an amazing, crystallography is an amazing field. You get some kind of, so this is heme. You get the structure of a heme, diffraction pattern, which is translated by a technique known as Fourier transform. Fourier. Now, if you are good with math, you know what this means. Fourier transform, it gives you a structure of a heme with the iron inside. Iron, this is the iron that will bind to the oxygen, as we will find out. Oops. Iron 2+. It'll bind to the oxygen. And then there's the pyrrole residues, iron with a heme, and finally, three-dimensional structure of the protein known as myoglobin. Myoglobin. Now, for the NMR, NMR is a different method. Again, there are a lot of new developments in this. So again, you have to purify the protein. You don't want any peaks which will contaminate your protein and give you wrong information. So you have to dissolve the protein. In most cases, this will be water, right? So the proteins live in water. And so can you dissolve this protein in H2O? In water?
No, no.
What will you use to dissolve the protein?
D2O.
D2O, perfect. You will dissolve in D2O. So remember, you use deuterated solvent for NMR because hydrogen will produce a very strong signal and which will overlap with everything else and will not let you visualize the actual molecule. So you have to use deuterated solvent, collect NMR data, assign, calculate structure. No need to crystallize, can see many hydrogens, all good things, but difficult for insoluble proteins. Again, bad for membrane proteins. So membrane proteins are a real difficulty. That's why I told you, if you get a structure of a membrane protein, especially something that people have been playing with for a long time, you will get a paper in Nature or Science. And if you get a paper, if you get the paper as a student, postdoc, if you get the Nature paper as a student or postdoc, then wherever you go after that, all the doors will be open for you. Right? Go do research, publish a paper in Nature, and then you apply to any medical school. Any medical school will accept you with open arms. Any graduate school will accept you, right? They will think that basically you're one of the top researchers the world has ever seen. And if you're top researcher, then you obviously, you know how to think. You will also be good with classes and be a very good doctor. Because most of the time with the doctors, it's not so much about looking up medication on Google, right? It's more about putting together different symptoms and figuring out what's wrong with you. All right. And so, so for the NMR, you can see that there are different techniques which are used. One of the most common which is used is this two-dimensional NMR. You see, these days, everything is multidimensional. We have advanced to the point that one dimension doesn't work for us anymore. So this will be a 2D NMR. And specifically, this one is known as a nuclear Overhauser effect, NOE, or sometimes referred to as NOESY. Now, I will not write down the whole thing for you, but you can Google it, you can read it in a textbook. Nuclear Overhauser. So the person who actually came up with this, his name was Overhauser. So basically what this tells you, this gives you the proximity, the signal, if you see a signal here, and then they have diagonal here, right? So the two hydrogens which are found on the diagonal here and there, one is 4.0 and one is 7.0. So you know these two hydrogens at these two ppm values are close in proximity. You know they're close in proximity, so they'll be interacting with each other. So if you have an alpha helix, right? You have an alpha helix, let's say, and let's say your two proteins are in side chains in which are from amino acids which are in position n and n plus 4, or n plus 5, as correctly pointed out to me several times by several of you. So n plus 5 works as well. So n plus 4 or n plus 5, then you know that these two hydrogens are in close proximity in the helix, and they might be on amino acids n and n plus 4. So same thing with this guy, hydrogen 2, right? So this one is slightly different. I'm not quite sure. This is heme. I think this is heme. This is heme. There's a methyl group here. There's, if you look at this blue hydrogen, this blue hydrogen, this blue hydrogen, they're all the same because the methyl group rotates about its axis, and they're all interacting with that hydrogen over there. And so that's how you would interpret the NMR data. So, but there are obviously many, many other possibilities. Any questions? All right. So let's see where we are.
I have one question about the last slide.
Yeah.
So the line that goes like, and like linear from one corner to the other, what does it mean if points are like, I guess in relation to that line, do points like, are they more or less close together, or what's the significance of that line?
Which line? This?
No, the diagonal one that goes
Oh,
like up to the right.
diagonal. Diagonal is basically is 10 to 0.
So
That-
what does it
that-
mean if points fall on that line?
You mean these? What's the significance of these ones?
Right, compared to the ones like point 1 and 2 where they're significantly off of the line.
Right. So in general, I can tell you when I use NOE, nuclear Oberhauser effect, this is something that is not exactly, because there's so many amino acids, there are so many dots here, right? It's not really easy to see, but if they found some specific interaction, which is the interaction between this hydrogen at 7 point whatever it is, 7.1, let's say, and this hydrogen 4.0, right? There must be another one at this position, because oftentimes two-dimensional NMR gives you some interesting data that is oftentimes you have to run it several times before you get convinced that the dot is actually there. But if you see it on both sides, right? If you see it on this side, and if you see it on this side, because you can see that
So it's like a line of symmetry.
line of symmetry, correct. But line of symmetry. So, okay, I think I overdrew it. It should be here. So, hold on a second. It should be, so if it's 4.0 here, it should be at 4.0 there. And 7 here, like that. Like that, right? So in other words, you will only be convinced that this point here is correct if you see the equivalent one on the other side, right? It's just basically your confirmation of the technique. Make sure the technique actually works. If you see the symmetrical point on the other side of the diagonal, then you say, okay, that's the true point, right? It's not something that that I picked up by mistake, that the instrument picked up by mistake. I mean, that's the only significance of this diagonal that I have had to use. So yeah.
Okay.
Make
Thank you.
sense?
Yes,
Yeah.
sir.
Yeah. So in other words, you can see what you can see here, right? So if there's a square here, it has to be symmetrical on both sides of the diagonal. Any other questions? NMR, X-ray. All right. All right. So proteostasis, stasis, whatever you call it. So so basically this is maintenance of cellular protein activity. That's the So basically, we have to make sure that all our proteins in the body are maintained in their native state, right? And so there's a variety of ways to accomplish that. Bodies do that. So the proteins are synthesized in the ribosome. We know that, right? So RNA comes in. Remember, RNA basically it's a copy of DNA. Now, before we start DNA and RNA, anybody remembers the difference between RNA and DNA?
The sugar backbone, DNA has deoxyribose and RNA is ribose.
That's right. And what's the difference between deoxyribose and ribose? In the structure, is there some particular structural feature? There's just only one functional group, right?
Deoxyribose has a phosphorus group, doesn't it?
They all have phosphorus groups.
Oh, okay.
Alpha helix in the DNA.
So this is
think
the
it's-
ribose. This is the ribose. Well, this is
Deoxy.
This is deoxyribose. What does ribose have?
I think it has OH versus deoxy has the just H there
Right.
in
This
the
is,
2.
yep. So this is 1 prime, 2 prime, 3 prime, 4 prime. That's right. So DNA will carry the information in the deoxyribose state. So this will be nucleic base. We'll talk about that when we get to it. And this will be the ribose, the hydroxy group. 1 prime, 2 prime, 3 prime, 4 prime. All right. So the RNA, which looks just a DNA only without this 2 hydroxy group, will arrive in the ribosome. What's transfer RNA? What does that do? Let's see if you remember some basic biology.
You said what did tRNA? Talking about the transfer.
Yeah, what's tRNA? What does it do?
Transfers amino acids.
Does
Right.
okay.
Yeah, it'll bring the
Not
amino
like I was
acid.
talking or anything.
Yeah. So it'll bring the amino acid. So the ribosome basically will accept the messenger RNA, right? So this will be the let's say mRNA. And there is also tRNA. Can't forget about that. Transfer RNA. So these are the key players in the ribosome. They will come in here, and their message will result into the protein nascent polypeptide. Now, nascent polypeptide, so there is this folding intermediate, right? So this folding intermediate is where things happen. So let me actually write some things down. Let's see what time I have. I still have plenty of time. So let me show and insert new slide. Don't need this. Something I love very much. It's called Levinthal's paradox. So if you have E. coli, bacteria, gram-negative bacteria, right? A lot of it lives in our gut. If you have E. coli, You know that E. coli is actively used in biotechnology, right, to synthesize proteins for us. Extremely prolific producers of proteins. Makes, when I say prolific, it's really prolific. It makes 100 amino acid peptide. In 5 seconds. What happened? What did I just do? What did happen to me before? One hundred amino acid peptide. In 5 seconds. At body temperature. At 37 degrees Celsius. But that's not enough, as we just learned. But protein making it is not the whole story. But protein must fold. All right. So assume now, if you look at amino acid, each amino acid can adopt, if you look at all these single bonds, each amino acid can adopt 10 different conformations. Conformations. Ten different conformations. And the protein tries out. Now, this is a huge simplification, right? So obviously there'll be more, but it's just approximation. Let's say 10 different conformations, but there'll be many more. Let's just limit ourselves to 10. Tries out each possible protein tries out each possible conformation before it finally folds. And let's say another number. Each conformation is sampled What's the length of a molecular vibration? Anybody remembers? So when you have a molecule of two atoms,
Is it
how
10 negative 13?
beautiful, beautiful. Within 10 to the negative 13 hours?
Seconds.
Seconds, nice. So 10 to negative 13 seconds. So this is molecular vibration. Oh. Molecular vibration. So if that's the case, then the calculations show it will take ten to the 77th years to sample all possible conformations. Clearly, that's not what's going on. All possible conformations. So there must be shortcuts. So the scientist who actually pointed this out, his name was Levinthal. Levinthal, one of my my most favorite principles in biochemistry, and known as Levinthal paradox. Science is full of paradoxes. Levinthal paradox. So clearly, this is not what happens. And so let's look at some of the major issues with denaturing. So protein function depends on three-dimensional structure. We talked about that. So loss of structural integrity is accompanied with loss of activity. Proteins can be denatured by heat or cold. So we talked about that, right? Heat or cold. We talked about high fever, right? Don't do that. Don't get overheated. Don't get too cold either. I don't know how. Can you get cold? Can you get I never thought about it. Has anybody thought about?
You can have hypothermia, which is when your body's temperature drops really low, generally due to outside forces.
Yeah, so like say if you go, let's say on a hike, right, and you get stuck, you get stranded at night at cold temperatures. Will you, when it's hypothermia, so your body temperature will drop?
Basically, it happens, yeah, generally because of outside forces. Like if you're outside in very, very cold weather and your body no longer can handle your body temperature regulation.
All right, I guess the same thing. So you will probably struggle or die from protein denaturing, right? Yeah, okay. pH extremes, so we talked about that as well, right? So if you especially have some ionizable side chains, which I will bug you on this test, get ready for that. Ionizable chains, right? So protonation, deprotonation, glutamate, aspartate, lysine, arginine, histidine, right? What happens with different pHs? Get ready for that. I'm gonna be bombard you with questions like that. And so obviously you change the charge, you will change the charge, as you change the charge, you change the shape, right? This can happen on the outside, right? So if you had all these charges on the outside and then all of a sudden all these amino acids become protonated, they go inside, or this can be a salt bridge inside the protein, you can break up with the pH as well. Organic solvents, remember organic solvents, if you move the protein from the aqueous phase to an organic phase, you can actually switch it inside out. It's like switching, you have a shirt or sweater and you turn it inside out, right? All the organic residues, hydrophobic residues that were inside, all of a sudden want to be on the outside because you expose the solvent to organic solvents. And agents such as urea, guanidinium hydrochloride. You remember from sapling, there was a reason why I gave urea question on the sapling. How many bonds, how many hydrogen bonds can urea make? Lots. That's why urea is used for protein denaturing. So here is you can see that percent unfolded protein versus temperature, right? You can see here for the enzyme ribonuclease A, and you can see here that actually it happens pretty quickly, right? So once you unfold, so for a while it stays okay, but then once you unfold a couple amino acids, it shoots straight up, right? So once you unfold a couple amino acids, the rest follow. So what's important to learn from this curve is that the folded state is very fragile. It's very fragile. It depends, oftentimes depends on just a few critical interactions. If you break those, you break the whole thing, right? And just in a few degrees, you're all the way up there from 0% unfolded to 100% unfolded. And so this is unfolding based on the temperature. This is unfolding due to the addition of guanidinium hydrochloride, hydrogen bonding breaker. And so this is ribonuclease refolding experiment. So ribonuclease, a small protein, contains 8 cysteines, and mercaptoethanol is a very useful small molecule reagent to break hydrogen bonds and to fully denature it. So the experiment quite simple, but it's so important it earned Chris Anfinsen 72 chemistry Nobel Prize. So you can see here that here's ribonuclease in catalytically active state. And remember it had 8 cysteines, right? So it has, you can see here, there's this yellow and black bonds, right? There are 4 of them between 4 cysteines. Now you can add urea and mercaptoethanol. You'll break all these disulfide bonds to release all these sulfhydryl links. Now you remove those and it spontaneously closes right back, which basically tells you that that the ribonuclease A did not need anything other than its own three-dimensional structure to reclose, to reform all these disulfide linkages. So disulfide linkages basically they reinforced the tertiary structure, but they're not required in this enzyme to adopt the three-dimensional structure ready for adoption of this three-dimensional tertiary structure. So very useful experiment that tells you that proteins, I think you have that in a sapling, that proteins by themselves do not require any assistance. Not all of them, but many do not require any assistance from the outside. But some of them. So here's another possibility. So let's say you have a protein which was just synthesized in the ribosome, and what will happen is that these segments, this segment will fold together to form into antiparallel beta sheet. This segment will fold together into alpha helix, and this segment will fold into antiparallel beta sheet. Then these will combine together, okay, into a motif, and then these will combine together, antiparallel beta sheet, and these two combine together into this motif. So you can see that the path to folding is actually, it's not just like one amino acid at a time, right? So the protein usually folds into these secondary structures first, which then come together and find their way to associate with each other. So protein folding in many cases is actually, it's a local process, right? So the several amino acids, maybe 15 or 20 of them, recognize their proximity next to each other, and they will fold into a secondary structure. This will happen somewhere else. It will happen somewhere else. And then those will bend towards each other, recognize each other, and come to form a motif, and so on and so forth, right? So this is how Levinthal's paradox can be overcome due to this stepwise process where the local folding precedes the more macro scale folding. All right, so this Levinthal's paradox we just talked about. And so this is another way to explain the Levinthal's paradox. It's sort of kind of known as a funnel model. Funnel model. So basically, you have, you can see the protein starts on the outside of the funnel, where the interactions between segments is very weak. But then there's some kind of favorable force which will force them into this funnel. They may come out, but this force is still stronger for them to come in. Okay, they go back in and out, in and out, in and out until they realize being inside is more favorable. And as they keep drawn further in and in and in and in, then the forces for them to stay inside are stronger and stronger and stronger. And then once they get all the way inside, then they realize this is where I want to be. This is where I want to be, and I don't want to come out, right? So this is the funnel model. And you can see here that actually the advantage of this is that they don't need to fully sample. So the conformations that they adopt here, they don't need to fully sample them. They can just kind of feel them, sense them. They like sort of, okay, maybe that's a good one, maybe not. Let me, out of maybe these 10,000 conformations, let me sample one, see if it's good enough or not. Okay, it's good. Okay, maybe let me make one more step towards the center of the funnel. Not good. Let me get out and make a different and go to a different funnel, right? And so, so you can look at this slide. Makes very good sense to me. And so the last things I want to mention to you. So there are proteins known as chaperones. Chaperones which assist in protein folding, right? So whose function is to assist in protein folding, and they prevent misfolding. So here's the folding intermediate, and there is chaperone HSP, known as heat shock protein 70, which will actually bind to the folding intermediate, right? Comes in and bind to it and will help the native protein find its native conformation. So oftentimes there are several of them. So there's heat shock protein 70, there's heat shock protein 40. So actually you can look at this. This is actually figure 430. You can read the legend. It's pretty self-explanatory. Now there is another family of proteins known as chaperonins. Chaperonins, so here is the, you can read about them, figure 431. Right, so it's almost like a jar, right? So your protein goes inside, and there's a cap. There's a cap, then you cook it inside, the cap comes off, and the protein comes back out. So very interesting family of proteins that perform this kind of folding. And what else do I need to mention to you? Diseases, obviously, you know about Alzheimer, right? Alzheimer, one of the most infamous Alzheimer's. So this is proteins which or the aggregates known as amyloids. Due to their hydrophobic nature, they aggregate in the brain, and so there is also transmissible ones. There's, for example, mad cow disease, right? So bovine encephalopathy. Let me see. A spongiform encephalopathy. BSE, sorry. So you can get them from eating steak, right? Don't get your steak.
Does Kuru, I'm sorry, does Kuru fall in this same like category, other pyron diseases?
Which disease, sorry?
Kuru, which is another,
Kuru. Yeah.
It's another pyron disease.
Yes, Kuru, prion. Did you mention prion? So prion disease is really bad because they're transmissible, right? So these are not only just proteins which are misfolded, they're actually infectious, right? They can go to brains of other people and make their proteins misfolded, right? So there's a lot of research in prion diseases. Obviously right now with COVID, I mean, research on any other kind of infections has gone down, not funded very well. But prion diseases are still some of the most challenging areas to investigate. So we still don't understand a lot about these prions and how specifically they make how these infections are transmitted from one patient to another. Kuru, prion. There's a sheep, scrapie.
Another one that's come to light is chronic wasting disease in deer, because people are hunting these deer, and we're concerned that it may be something that can be passed on to people, but we're not sure yet. There's still been ongoing.
You said chronic
wasting disease. Mm-hmm.
Yeah. So protein folding is a huge area, wasting disease. I mean, that's why we spend out of what, 12 lectures or 13 lectures on this class, we spend almost the entire, sorry, not lectures, chapters. we spent the entire chapter on protein three-dimensional structure, right? Because I mean, that's the beauty about proteins, is that they work. I mean, primary structure determines their secondary and tertiary structure. And if it was just the primary structure, then it would be not sufficient to understand how proteins work. You really need to know what they look like in 3 dimensions, what kind of pockets they form. If you develop a drug, if you discover a drug, you know, if you're a medicinal chemist working for a big pharma company, you decide, say, you know, you get a degree in biochemistry, biology, or chemistry at Texas State, and you want to work for a pharma company, make some good money. Pharma companies pay a lot. So you know, you're gonna look in your project manager is gonna come to you and say, you know, we just discovered that this protein is responsible for chronic wasting disease, and you know, we want to have a drug for it. And so the market is, I don't know what the market is for it right now, but there's nothing out in the market. We're gonna dominate the market if we have a drug for it. And here is the primary structure. So what do? Well, you can't have a primary structure and all of a sudden develop a drug, right? So you have to go and actually understand what this protein looks like in 3 dimensions, right? You have to understand what kind of secondary structures, what kind of tertiary structure, what kind of pockets, how it works, what kind of drug, where it is going to go, right? Understand various types of intramolecular interactions. Are there going to be hydrophobic forces? Are there going to be salt bridges? Are there going to be pole interactions, and so on and so forth? Are there going to be other sites on the molecule, maybe regulatory sites, right? So there's enzymatic site, but there must be a regulatory site where the drug might bind and slow down the enzyme as opposed to kill it right away. So, all right. So guys, hopefully this was useful. I would like to see more comments on the discussion. And on Wednesday, we're going to start chapter 5. And we're going to follow that with the first test on next Wednesday. Any questions?
I have a quick question. Somebody, I think it was Riley, maybe mentioned doing a Zoom meeting to study. I just wanted to bring it up while people are here. If that's something that people are interested in, maybe we can post a Zoom meeting for people to join. I think Riley was mentioning like this weekend. I just wanted to put it out there so that everybody was aware that wanted to attend.
Sure. You guys do whatever works for you. I mean, I prefer study by myself, but if you like to study in groups, that's fine too. But look, guys, you should be, when you study like that, you should be studying already kind of now and not leaving it a few hours before the exam. That's just my recommendation.
I was gonna ask about the format. Is the test gonna be the same format as the sample exam, or is it gonna be different?
Yeah, it's gonna be the same. Mm-hmm.
So on Canvas?
Yeah. Mm-hmm.
Okay, thank you.
It's gonna be 30 questions in 50 minutes, but the question is gonna be just like this. So
okay, so almost choice basically.
For many questions, you will probably take you like a few seconds to answer. So
thank you.
Yeah. All right. Any more questions? Just put in discussion or email me.
Will the test start at 11 o'clock? Does it have a time, like certain time that?
Yes, the test will start sharp at 11 o'clock and go to 11:50, 50 minutes. Mm-hmm.
Okay, thank you.
Mm-hmm. Yeah, don't be late. Okay. Talk to you